Usually when we talk about a medical diagnosis, there's this expectation of precision.
Oh, absolutely.
It's almost like engineering. Like
Yeah.
you fall off a bike, you break your arm, the X-ray shows that jagged white line on the screen,
Mhm.
and the doctor just points at it and says, there it is. That's the problem.
Right. It's a very comforting way to view the human body. I mean, we crave that binary simplicity,
Yeah.
broken or not broken, infected or clear. We want our health to be visible and easily categorized.
Exactly. But the moment you step out of the ER and into the world of like cellular biology or aging or chronic disease, that X-ray machine suddenly becomes completely useless.
Well, completely. You're looking at a microscopic landscape that is, well, infinitely more complex than a fractured bone.
Right. But today, we're going to explore a cellular survival mechanism that is happening inside you right now. Like literally, as you listen to this deep dive.
It's
It really is. It's an innate, highly efficient housekeeping system capable of literally cannibalizing its own damaged parts to reverse aging, clear out disease, and completely renew your biology from the inside out.
I mean, it sounds like something out of a science fiction novel, right?
Literally.
The concept that our own cells can consume themselves to ensure our survival. But understanding how to control this mechanism specifically through when you eat and by utilizing certain ancient plant compounds is arguably the most powerful tool we have against the diseases of aging.
So today, our mission is to take you on a journey. We're going to start down at the absolute microscopic level of cellular renewal, examining the literal architecture of a cell.
Uh-huh.
And we're going to follow that thread all the way up to your daily practical wellness. We're looking at the cutting-edge molecular biology of a process called autophagy, exploring the clinical protocols of the fasting mimicking diet, and bridging all of that with 5,000-year-old Ayurvedic wisdom regarding, the chemical power of specific spices.
It's this brilliant intersection of modern clinical data and ancient human habit.
Okay, let's unpack this.
Mm.
Because before we can understand how to hack our health with fasting windows and turmeric, we have to zoom all the way in.
Right.
We need to see the architecture. What is actually happening inside a single microscopic cell when this process kicks off?
Well, we have to start with the term itself, right? Autophagy. If we look at the literal translation from the Greek, auto means self and phage means eat.
Self-eating.
Exactly. It translates directly to eat thyself. And in biological terms, it is a critical homeostatic mechanism.
And homeostasis is just a fancy way of saying, like, the body's trying to keep the house in order, right? Trying to maintain a stable, balanced environment.
That is the core of it.
Yeah.
Inside every single one of your cells is this jelly-like substance called the cytoplasm.
Okay.
And floating in that jelly are essential components called organelles. You can think of organelles as mini organs. Just as your body has a heart to pump blood and lungs to process oxygen, your cells have specific organelles to produce energy, build proteins, and manage waste.
That makes sense.
But over time, due to stress, toxins, or just normal cellular metabolism, these mini organs get damaged. They start misfiring.
Like parts wearing out in a car engine.
Precisely. Autophagy is the process where a cell actively digests portions of its own cytoplasm and these broken organelles for removal and turnover.
It's incredible to think this was largely mysterious until very recently. Like the Japanese cell biologist Yoshinori Osumi actually won the Nobel Prize in Physiology and Medicine back in 2016 for finally mapping out how this works.
Yes, his work was groundbreaking.
The scientific community knew cells could degrade their own contents, but Osumi proved that during periods of fasting, cells literally turn on specific autophagy genes.
His work with yeast genetics was the skeleton key that unlocked our understanding of mammalian cells. And the mechanism he illuminated is breathtakingly elegant. It all starts with a stressor. The most profound trigger for this entire genetic cascade is nutrient deprivation. Starvation.
Okay.
When a cell realizes the food supply has been cut off, it initiates a highly choreographed biological pathway.
So I want to walk through this pathway step by step because the mechanics are astonishing, but the terminology can feel like, alphabet soup.
Oh, definitely. It gets very technical.
Well, let's break it down. The body senses a lack of nutrients. What is the very first domino to fall inside the cell?
The initiation phase begins with a protein complex called the ULK complex. In yeast, it was called at G1, but in humans, it's ULK, which stands for UNC51, like autophagy activating kinase.
So if we imagine the cell as a massive manufacturing factory, this ULK complex is essentially the emergency alarm system on the factory floor that detects a supply chain failure.
That's a great way to picture it. It senses the lack of incoming energy, and once that ULK alarm is triggered, it enhances the activity of a secondary system, a complex known as the class 3 phosphatidylinositol 3 kinase complex 1.
Okay, wait, slow down. Phosphatidyl what?
Phosphatidylinositol 3 kinase.
Right, let's just call that the PI3K complex.
Fair enough.
What is this secondary system actually doing on the factory floor?
Well, the PI3K complex has one specific job. It synthesizes a highly specialized lipid, a fat molecule called PDDIN-S3P.
A lipid signal. So the alarm goes off and this system shoots a lipid into the cell. It's like firing a chemical flare gun inside the factory to call in the cleanup crew.
That is precisely what it does. The accumulation of this specific lipid acts as a glowing beacon. It promotes the expansion of cellular membranes and starts recruiting dozens of other autophagy-related proteins to a very specific location.
Okay.
All of this emergency activity converges on a specialized region of the cell's endoplasmic reticulum.
And the endoplasmic reticulum is the part of the cell that usually manufactures new proteins and fats, right?
Yes. But during starvation, a specific subdomain of it transforms. This staging area is called the omegasome.
The omegasome. I love that. It sounds like a sci-fi staging ground.
It essentially is the birthplace of the entire recycling machinery. At the omegasome, a temporary double membrane structure begins to physically manifest.
Wow.
In its early stages, it's called a phagophore. Under an electron microscope, researchers have watched it begin as a tiny, crescent-shaped sliver of membrane that slowly grows into a cup-shaped structure, maybe a micrometer across.
Okay, let's push the analogy further to visualize this.
Right.
Because the word eat implies a mouth, but obviously cells don't have mouths.
Right.
If the omegasome is the staging area in our factory, the phagophore is the actual cleanup crew. But instead of a guy with a broom, imagine a Pac-Man made entirely out of a highly flexible, double-layered balloon.
The flexibility is the key trait. It has to physically bend and contort around the debris.
Right, so this flexible balloon Pac-Man approaches a piece of toxic cellular garbage, maybe a broken protein or a dying mini-organelle. But instead of just taking a bite out of it, the balloon expands, wraps entirely around the garbage, and keeps extending until the two ends meet and seal shut, locking the garbage inside a completely separate airtight bubble.
The visual of the balloon sealing shut is chemically perfect. Once that sequestration step is complete, the structure changes names. It is no longer a phagophore. It is now a sealed double membrane vacuole called an autophagosome.
So it's a microscopic trash bag full of broken cellular machinery.
Exactly.
But it doesn't just sit there. Where does it go?
Well, the autophagosome is fundamentally a delivery vehicle. It navigates through the cellular jelly to find the ultimate recycling center, which is the lysosome. Lysosomes are pre-existing organelles packed with highly potent acidic degrading enzymes.
So it's the incinerator of the cell.
Or the acid bath. The autophagosome literally docks and fuses its membrane with the lysosome. They merge into a single entity called an autolysosome.
Oh, wow.
Inside this new chamber, those acidic enzymes are unleashed on the trapped cellular garbage. They break down the damaged proteins and dysfunctional organelles into their most basic elemental building blocks, individual amino acids, and free fatty acids.
And then the cell just reuses them.
Yeah.
It literally melts down the broken office chairs to build new tools and generate energy to survive the famine. That changes the whole way I think about hunger. It's not just an energy deficit, it's an active demolition and reconstruction phase.
Which is exactly why chronic, uninterrupted eating is so detrimental. You are never giving the factory the signal to melt down the broken chairs.
Wait, I have to challenge this a little.
Go ahead.
If a cell is full of broken organelles and toxic proteins, doesn't the body already have a mechanism to just kill that cell? I thought our bodies were constantly shedding dead cells and making new ones. Why go through this elaborate, energy-intensive process of internal recycling if you can just scrap the whole cell?
You're referring to programmed cell death, which is scientifically known as apoptosis.
Apoptosis, okay.
And you are correct, the body does use apoptosis to eliminate permanently damaged cells. But there is a massive biological cost to apoptosis.
What kind of cost? Like energy?
It is an incredibly messy and inflammatory process.
Yeah.
Think of apoptosis like demolishing an old building with dynamite.
Yeah.
Yes, you get rid of the building, but you are left with a massive cloud of toxic dust, debris, and shrapnel flying into the surrounding neighborhood. When a cell undergoes apoptosis, it ruptures and spills its metabolic waste into the surrounding tissue. This forces your immune system to rush in, triggering systemic inflammation.
So apoptosis is a desperate last resort.
Exactly. Autophagy, conversely, is highly energy efficient and perfectly clean. Because the degradation happens entirely inside that sealed, airtight bubble of the autolysosome, absolutely no toxic debris is spilled into the surrounding tissue.
That's amazing.
The more your cells can repair themselves through autophagy before the damage becomes critical, the less you have to rely on the dynamite of apoptosis and the lower your overall bodily inflammation remains.
Okay, that clarifies the stakes immensely. We want dismantling, not dynamite.
Right.
So if that is the microscopic process, the PI3K flares, the flexible phagophores, the lysosome acid baths, how does this actually manifest when we zoom out? Like, if I am a listener and I optimize this process, what happens to my actual organs?
To appreciate the benefits, we first have to look at the alternative. When this autophagy process slows down, which happens naturally as we age, but is massively accelerated by modern lifestyles, cells become damaged but fail to trigger either autophagy or apoptosis.
Oh, no.
They enter a state of limbo. These are known as senescent cells.
Senescent cells. In longevity circles, people always call these zombie cells.
It's a very apt nickname. They are decaying, dysfunctional cells that have stopped dividing, but they refuse to die. And worse, they aren't just taking up space. They actively secrete a cocktail of inflammatory chemicals and toxins.
Ooh, so they're poisoning the area.
Yes. They essentially infect the healthy cells around them, dragging the tissue into a state of chronic disease. Autophagy is your primary mechanism for hunting down and clearing out the molecular damage that creates these zombies.
Let's apply that to specific systems. Take the gut, for instance. We are living through an epidemic of digestive disorders right now.
The clinical data on intestinal health and autophagy is striking. The lining of your gut is incredibly thin, just one cell thick in many places, and it relies on tight junctions to keep food particles and bacteria from leaking into your bloodstream.
Right.
When autophagy is humming along in the gut lining, it is constantly recycling the damaged proteins in those tight junctions, literally rebuilding the cellular barrier in real time.
So a lack of autophagy directly contributes to leaky gut syndrome because the barrier is left to decay.
Exactly. Without the cleanup crew, the barrier degrades, pathogens slip through, and you develop severe gut inflammation. The same principle applies externally to the skin.
I imagine skin cells are highly susceptible to becoming those zombie cells because of, UV exposure and environmental toxins.
They are. High levels of regular autophagy clear out the cellular junk that degrades elastin and collagen. People who successfully trigger regular autophagy show vastly more resilient skin. We see clinical reductions in eczema and adult acne, and a significant slowing of visible aging markers like wrinkles, because the skin is constantly renewing its structural proteins from the inside out.
That makes total biological sense.
Yeah.
If you clean out the zombies, the tissue behaves younger.
Precisely.
But let's talk about the brain, because this is where the implications get deeply psychological and honestly a little scary when we think about neurodegenerative diseases.
The brain is the most metabolically active organ in the body. It consumes massive amounts of energy to process thoughts, manage the nervous system, and regulate emotions. But that high metabolic rate means it produces an enormous amount of cellular exhaust or metabolic waste.
The physical byproduct of thinking.
Quite literally. Autophagy in our brain cells, our neurons, is the only way to clear that exhaust. When the process is active, we see immediate psychological benefits. Better mood regulation, sharper memory retrieval, and quicker executive processing.
Wow.
The brain simply runs faster when the engine isn't clogged with debris.
And long term.
Yeah.
We're talking about conditions like Alzheimer's and Parkinson's.
Those diseases are fundamentally characterized by the accumulation of toxic protein aggregates. In Alzheimer's, it's amyloid beta plaques and tau tangles. In Parkinson's, it's Lewy bodies.
Right, you hear about those a lot.
These are misfolded proteins that clump together and slowly crush the neurons to death. Autophagy is the precise evolutionary mechanism designed to recognize those misfolded proteins, wrap a phagophore around them, and dissolve them before they can clump together.
So a failure of autophagy in the brain isn't just about brain fog. It's the direct precursor to dementia. That is wild.
It is.
And scientists have actually filmed this happening, haven't they? The data utilizing glowing mice.
The GFP-LC3 transgenic mouse model, it's a landmark achievement in observational biology. Researchers genetically engineered mice so that a specific protein related to the phagophore formation was fused with a green fluorescent protein.
The exact same protein that makes jellyfish glow in the dark.
Yes. By tagging the cleanup machinery with this fluorescent marker, they didn't have to guess if autophagy was happening. They could literally see it illuminating under a fluorescence microscope.
That is so cool.
They subjected these engineered mice to 24 hours of food withdrawal.
Just a single day of fasting.
And when they looked at the cryosections of the tissues, the results were undeniable. Massive systemic autophagy had been visually confirmed. The cells were glowing green across the skeletal muscle, the liver, the heart tissue, and notably the kidney podocytes.
Wait, what is a podocyte?
Podocytes are highly specialized, tentacle-like cells in the kidneys that wrap around the capillaries. They are the actual physical filters that clean your blood. They have a very high turnover of proteins. The fact that a mere 24 hours of starvation triggered profound autophagy in these critical blood filtering cells proves that this isn't just an obscure cellular quirk. It is the primary systemic software program for human survival and maintenance.
There was another mouse study that looked at this neuroimmune connection, specifically regarding diet, right? Comparing how high-fat versus low-fat diets impact the brain during fasting.
Yes, published via the National Center for Biotechnology Information. They took two groups of mice. One was fed a standard low-fat diet and the other a high-fat diet for roughly 12 weeks. Then, both groups were subjected to a fasting protocol to trigger autophagy.
And what happened to their brains?
The mice that entered the fast from a low-fat diet showed profound cognitive improvements. They vastly outperformed the high-fat group on complex memory and learning tasks. Their locomotor activity, their physical sharpness was elevated. The researchers found that the fasting exerted a powerful anti-inflammatory effect on the entire neuroimmune system.
But the high-fat diet blunted that effect.
In the specific parameters of this study, yes. The lipid overload seemed to interfere with the neuroimmune reset that fasting usually provides. It highlights that what you eat before you stop eating matters.
Okay, so we've established the stakes. This process rebuilds the gut, tightens the skin, filters the blood, and literally cleans the brain to stave off dementia. It is a biological superpower. But the obvious glaring question for anyone listening is, how do we intentionally turn this on? Because obviously chronic starvation isn't a viable lifestyle.
No, of course not. And that tension is exactly why modern society is drowning in chronic degenerative diseases. Heart disease, diabetes, Alzheimer's, autoimmune conditions. Our current medical paradigm often accepts these as an inevitable consequence of getting older.
But they are largely the consequence of us turning off the maintenance software, right?
Precisely. Evolutionarily, human beings were subjected to frequent, involuntary periods of famine. Our biology was forged in scarcity. We developed this incredible capacity to heal and repair ourselves during those periods without food. But our modern environment is one of absolute, inescapable abundance. The habit of round-the-clock eating breakfast at 7 a.m., snacking all day, eating right up until bed, completely thwarts this innate mechanism.
Because if the food is constantly coming down the conveyor belt, the factory never sounds the ULK starvation alarm. It just keeps trying to process the new material, letting the broken machinery pile up in the corner.
At a biochemical level, this dynamic is controlled by two primary competing genetic switches.
Let's map out the control panel. What are these switches?
The positive regulator, the switch that senses low energy and turns autophagy on, is called AMPK. It stands for AMP-activated protein kinase. When your cellular ATP, your energy currency, drops, AMPK senses that deficit and fires the signal to start the cleanup process.
So AMPK is the master switch for longevity and repair. What is the opposing switch?
The negative regulator, the switch that senses abundance and turns autophagy off, is the mTOR pathway, specifically mTORC1. mTOR stands for the mechanistic target of rapamycin.
Okay, I have to jump in here because if you spend any time in the fitness or bodybuilding world, you hear about mTOR constantly. Bodybuilders want to spike mTOR because it drives muscle growth.
They do. mTOR is the master anabolic signal. When it senses an abundance of nutrients, particularly amino acids from dietary protein, it tells the cell, times are good, synthesize proteins, multiply, grow muscle.
Makes sense.
But here is the biological catch. When mTOR is highly active, it forcibly suppresses AMPK and completely shuts down autophagy.
So they are fundamentally at war. You cannot be in high-growth, muscle-building mTOR mode and deep-cleaning, anti-aging AMPK mode at the same time. It's biologically impossible.
It is a seesaw. You cannot have both ends up at once. To achieve optimal health, you must oscillate between the two. You want MTOR active sometimes to maintain muscle mass and bone density, which is critical for longevity.
Right.
But the modern crisis is that the average person is stuck in a state of chronic 247 MTOR activation. The seesaw is permanently jammed in the growth and multiply position, which eventually leads to cellular senescence and tumor growth.
So how long does it actually take to unjam the seesaw? Like, if someone puts down the fork, what does the biological timeline of fasting actually look like?
The first major shift happens around the 12-hour mark.
So just finishing dinner at 7 p.m. and not having breakfast until 7 a.m.
Right. At 12 hours, you've generally depleted your glycogen stores, the reserve sugar kept in your liver, and your body steps into a fat-burning state. This is often called circadian fasting. It aligns your digestion with your circadian rhythm, giving your gut a baseline rest.
Okay.
It's highly recommended for daily health, but it doesn't trigger the deep systemic autophagy we are talking about.
It's just sweeping the floor, not melting down the chairs. How far do you have to go for the deep clean?
The clinical data points to the 48-hour mark as the threshold for profound transformation. After 48 hours of zero caloric intake, the body firmly flips the genetic switch from growth to repair. AMPK is fully dominant. Autophagy kicks into high gear across the major organ systems.
2 full days without food. That is a massive physiological shift.
It is, and the effects on the immune system alone are revolutionary. For example, at the 48-hour mark, autophagy specifically begins targeting and recycling overactive immune cells.
Wait, if there are pathogens everywhere, why would our body want to recycle its own immune cells? Don't we need those?
We need healthy, calibrated immune cells. We do not want dysfunctional, hyperactive immune cells. Consider mast cells, for instance.
Mast cells, those are the ones that burst open and release histamine when you have an allergic reaction, right?
Exactly. They are the alarm bells of the immune system. But in millions of people suffering from conditions like histamine intolerance or MCS, mast cell activation syndrome, those mast cells are damaged and hyperactive. They are constantly degranulating, flooding the body with histamine, causing chronic hives, severe brain fog, digestive collapse, and systemic inflammation.
The alarm bell is just ringing endlessly for no reason.
Yes. By pushing into that 48-hour fasting window, the AMPK pathway directs the phagophores to physically swallow and dismantle those hyperactive mast cells. It essentially deletes the faulty immune cells, and upon refeeding, replaces them with a fresh, correctly calibrated batch. The reduction in systemic histamine levels is profound.
That is an absolute lifeline for people dealing with severe autoimmune or allergy issues.
It truly is.
But we have to acknowledge the reality here. Asking the average person, especially someone who is already sick, fatigued, and stressed, to just stop eating for 48 or 72 hours is an enormous hurdle. It spikes cortisol, it ruins sleep, and it requires Herculean willpower.
Which brings us to one of the most exciting developments in modern nutritional science. Researchers recognize the clinical impossibility of prescribing 3-day water fasts to the general public, so they engineered a biological workaround.
Here's where it gets really interesting. Tell me about the FMD, the fasting mimicking diet.
The FMD is a brilliant piece of biochemical deception. The goal is to grant you the profound disease-reversing benefits of a 5-day water fast, while actually allowing you to eat solid food.
How does that even make sense? If you are eating food, how does the ULK alarm system not go off?
It all comes down to the specific thresholds of those nutrient-sensing pathways we discussed, MTOR, as well as PKA and IGF-1, which also signal growth. The FMD is a highly calibrated, plant-based dietary protocol.
Okay.
The food is formulated to slip entirely under the radar of those sensors.
Like a stealth bomber evading the cellular radar.
Yeah.
What is the actual macronutrient breakdown that achieves that stealth?
To evade MTOR and IGF-1, the diet must be exceedingly low in amino acids protein and exceedingly low in simple sugars, because protein and glucose are the primary triggers that jam the seesaw into growth mode.
Right.
Instead, the FMD provides precise amounts of healthy, plant-based fats and complex carbohydrates.
So you are consuming enough calories to prevent extreme cortisol spikes, maintain your basic metabolic functions, and keep you sane. But biologically, your cells are convinced you are starving in a desert.
Precisely. You remain below the nutrient-sensing threshold. Let's look at the timeline, which typically spans 5 days.
Walk me through the 5 days. What is happening internally as you eat this stealth food?
Days 1 and 2 are the transition phase. You are gradually shifting the body's fuel source. You burn through the circulating glucose and enter a deep state of ketosis, where the body begins breaking down visceral fat to produce ketone bodies for brain fuel.
So the first 2 days are just changing the gas tank. When does the cleanup start?
Day 3 is the pivot point. This is when the cellular environment registers the sustained lack of protein and sugar. AMPK surges and elevated autophagy begins. The omegasomes start churning out phagophores. Days 4 and 5 are where you reap the compounding rewards. By maintaining the stealth diet, you cultivate a profound state of cellular rejuvenation. The body is actively breaking down visceral fat, recycling senescent cells, dismantling faulty mast cells, and clearing amyloid plaques.
And all the while, you're eating soups and nut bars.
Yeah.
It takes the suffering out of the equation while delivering the clinical biology. It's genius.
It democratizes the benefits of extended fasting.
Okay, but let's say you complete your 5-day FMD, or you do a traditional 48-hour water fast. You feel incredible. Your brain is sharp. Your joints stop aching. But eventually, you have to go back to eating a normal diet.
Right.
The seesaw has to tip back to MTOR eventually. When we do eat, how can we prevent all that inflammation from just rushing back in? How do we keep the momentum going?
This is where we bridge the gap between cutting-edge molecular biology and 5,000 years in traditional wisdom, because you can actually sustain a low level of AMPK activation, a daily hum of autophagy through the specific chemical compounds found in certain plants.
We are opening the ancient spice cabinet.
Exactly. When you examine the chemical structures of certain spices, you find a high concentration of molecules called polyphenols. These are powerful plant-based antioxidants, but they don't just passively neutralize free radicals, they are active signaling molecules.
Are you saying they interact with the seesaw?
Directly. Specific polyphenols chemically mimic the start of fasting. When they enter the bloodstream, they actively bind to the cellular machinery to suppress the MTOR pathway and upregulate the AMPK pathway.
So eating these spices literally tricks the cell into thinking it's fasting, even when it isn't.
To a certain degree, yes. It isn't as blunt a force as a 3-day water fast, but when used in conjunction with a daily 12 to 16-hour fasting window, these spices act as a massive amplifier.
Well, that makes sense.
The science shows a synergistic effect. The fasting window primes the cellular receptors, making them vastly more sensitive and responsive to the polyphenols when you finally do eat.
Okay, let's get specific. What are the heavy hitters in this polyphenol world? The sources highlighted a few, but the history on ginger really caught my attention.
Ginger is a biological powerhouse. It is a rhizome and has been a foundational pillar of Ayurvedic and Chinese medicine for over 5,000 years. It was so highly valued that the Romans imported it extensively to Europe for medicinal use.
5 millennia of human trial and error, now validated by electron microscopes. What makes it so chemically potent?
A single ginger root contains over 100 distinct chemical compounds. But the molecular superstars responsible for the health benefits are called gingerols and shogaels, specifically 6-shogael.
6-shogael. It sounds like a synthetic pharmaceutical, but it just grows in the dirt. How does it work?
Ayurveda traditionally refers to ginger as a digestive fire, believing it burns away toxins. In modern biochemical terms, 6-shogael is a direct AMPK activator. It signals the cell to initiate autophagy.
Wow.
But a most immediate clinical impact is on pain and inflammation. 6-shogael actively blocks the inflammatory pathways in the nervous system.
The text specifically highlighted its clinical efficacy in reducing the pain associated with exercise-induced muscle damage, osteoarthritis, intestinal cramping, and even the systemic aches of metabolic syndrome. It's essentially a natural NSAID, but instead of damaging your stomach lining like ibuprofen, it heals the gut-tight junctions.
The versatility is remarkable, and it works synergistically with its famous botanical cousin, turmeric.
Turmeric. The bright yellow root, I feel like this has been the trendiest supplement for the last decade, but the actual volume of data on it is staggering.
It is arguably one of the most rigorously researched botanical compounds on Earth. The U.S. National Center for Biotechnology Information alone has cataloged over 6,000 peer-reviewed studies on turmeric and its primary active compound, curcumin.
How does curcumin fit into the autophagy puzzle?
Curcumin is a profoundly potent polyphenol. Similar to 6-shogael, curcumin has been shown in vitro to aggressively suppress the MTOR growth signal while upregulating AMPK. It chemically forces the cell to prioritize cleanup over growth.
So ginger and turmeric are the kings of the spice cabinet. What other plant compounds should we be looking at to keep this daily cellular maintenance running?
The list is surprisingly accessible. Another major polyphenol is quercetin, which is found in high concentrations in the skins of red onions, cranberries, and elderberries.
I've seen quercetin used a lot in allergy formulas.
Because it is a phenomenal mast cell stabilizer. We talked about how fasting dismantles broken mast cells. Quercetin acts on the remaining mast cells, physically stabilizing their membranes so they don't rupture and dump histamine needlessly. It's a perfect complement to the fasting mechanism.
That is a brilliant combination. What else?
There is carnosic acid, which is abundant in savory, woody herbs like oregano, sage, and rosemary.
So heavily seasoning your meals with rosemary isn't just for flavor. You are literally dosing yourself with AMPK activators.
Precisely. Then we have EGCG, a highly studied catechin found in green tea, oolong tea, and dark chocolate. There is citrus bergamot, the essential oil that gives Earl Grey tea its distinct flavor profile. And finally, resveratrol, the famous polyphenol found in the skins of dark berries, grapes, and red wine.
Okay, so we have this arsenal of compounds. The text actually provided a highly specific protocol for putting this into practice immediately. A lemon ginger tea recipe designed to maximize the extraction of these polyphenols. I want to share this with you listening because it's such a practical, immediate takeaway that you can do in your kitchen today.
It is an exceptional way to break a daily fast or to consume after a meal to aid the digestive process and keep inflammation low.
Here's the exact botanical extraction protocol from the text. Step 1. Take a 3-inch piece of fresh, raw ginger root. Peel the skin off and slice it as thinly as possible to maximize the surface area. Step 2. Put those slices into a pot with 2 cups of water and bring it to a boil for 10 to 15 minutes, making sure the lid is firmly on.
The lid is non-negotiable. Many of the most potent chemical compounds in ginger, including certain gingerols, are volatile oils. If you boil it without the lid, those compounds literally evaporate into the air of your kitchen. The lid forces them to condense and drip back into the water.
That makes perfect sense. The text also notes a matter of physics. Boiling it for 10 to 15 minutes will cause you to lose about half the water volume to evaporation, resulting in a highly concentrated tea. If you want to make a large batch for the week, say using a 6-inch piece of ginger, you need to start with 3 to 4 cups of water and boil it for a full 30 minutes.
And that extended boiling time is actually crucial for the chemistry. The sustained heat slowly converts some of the gingerols into that highly potent 6-shogol we discussed earlier.
So you are actively changing the chemical structure in your kitchen. Step 3. Turn off the heat, remove the lid, and let it cool. Step 4. Once it is warm but drinkable, pour it into a mug, squeeze in a quarter of a fresh lemon, and if you need it sweetened, add 3 to 4 drops of lemon drop stevia.
The additions are very intentional. The lemon provides a hit of vitamin C and citric acid, which supports liver detoxification. And utilizing stevia instead of honey or sugar ensures that you do not spike your insulin.
Ah, right.
If you add sugar, you instantly trigger the mTOR pathway and shut down the autophagy we're trying to promote.
It is basically a warm, delicious mug of cellular maintenance. That's right. We have covered an immense amount of ground. We started with the microscopic architecture of the ULK complexes and lysosomes. We've traced the psychological benefits for the brain, the biology of the FMD, and the chemical mimicry of ancient spices. Let's tie all these threads together in this final section.
This is good.
If someone adopts this lifestyle utilizing fasting windows and these specific spices, how does this fundamentally alter their trajectory regarding long-term disease, energy, and physical performance?
To connect the final dots, we have to look at the root driver of almost all modern pathology. We have mentioned systemic inflammation, but we need to understand how it destroys the body.
What is actually happening in the blood when we are inflamed?
At a molecular level, your immune system uses signaling proteins called cytokines to communicate. 2 of the most aggressive pro-inflammatory cytokines are interleukin-6, or IL-6, and tumor necrosis factor alpha, or TNF-alpha.
Okay.
When you have a cut or an acute infection, these cytokines spike to orchestrate the healing process, and then they disappear. But due to our diet's toxins and lack of autophagy, the average modern human has chronically elevated levels of IL-6 and TNF-alpha.
The alarm is just blaring 247.
Yeah.
And that constant state of biological panic is what degrades our tissues.
Exactly. It causes the immune system to lose its precision. And this is where the synergy of our discussion is so clear. Both intermittent fasting and the daily intake of polyphenols like curcumin and 6-shogol have been clinically proven to actively suppress the release of IL-6 and TNF-alpha.
They literally reach into the control panel and turn down the volume on the inflammation alarm.
They do, and this is precisely why individuals suffering from severe autoimmune diseases where the immune system attacks the body's own tissues, see such remarkable turnarounds with fasting and spice integration.
That's incredible.
Conditions like multiple sclerosis, rheumatoid arthritis, Crohn's disease, colitis, and systemic lupus are all driven by this misdirected cytokine-fueled fire. Fasting starves the fire, and the spices act as the chemical water, allowing the immune system to finally reset and normalize.
Speaking of the immune system, take that logic one step further. If autophagy is this incredible Pac-Man that eats broken proteins and dead organelles, what happens when a virus enters the cell? Does autophagy fight infections?
This is perhaps one of the most elegant adaptations in human biology. There is a highly specialized targeted form of autophagy called xenophagy.
Xenophagy. The prefix xeno means foreign or alien.
Exactly, the eating of foreign invaders. During a fasting state, when AMPK is high, the body doesn't just look for its own broken parts. It actively scans the intracellular environment for non-self entities invading viruses, pathogenic bacteria, and parasitic yeast.
Wait, are you saying the Phagophore balloon can literally wrap itself around a live strep bacterium?
Yes, it identifies the pathogen. The phagophore engulfs the live bacteria, seals it into an autophagosome, and then docks with the lysosome. The acid bath dissolves the invading germ completely. Xenophagy is a primary innate pillar of your immune defense.
This completely recontextualizes how I think about being sick, because what is the very first thing that happens when you get a bad flu or a fever? You completely lose your appetite. The thought of food makes you nauseous.
And for generations, well-meaning mothers and doctors have told us to feed a cold, starve a fever, or just to force ourselves to eat to keep our strength up.
But the loss of appetite isn't a symptom of the illness, it's an evolutionary strategy. Ayurveda has recognized this for thousands of years, recommending strict fasting during fevers. The body is intentionally suppressing your hunger hormones because it desperately wants to keep MTR turned off and AMPK turned on. It is trying to maximize xenophagy so it can actively hunt and digest the virus.
Precisely. When we force ourselves to eat a bowl of sugary chicken noodle soup because we think we need energy, we spike our glucose, trigger MTOR, and instantly shut down the xenophagy machinery. We are accidentally turning off the very mechanism designed to destroy the pathogen, which often prolongs the infection.
That is profound. It requires you to completely trust the innate intelligence of your body. Let's pivot to the final major system the source is detailed, our actual metabolic energy. How does autophagy interact with our blood sugar and our daily energy levels?
Everything regarding human energy comes down to the health of your mitochondria.
The classic high school biology answer, the powerhouse of the cell.
It's a cliche for a reason. Mitochondria are the unique, complex organelles responsible for producing ATP, cellular energy. A pristine, healthy mitochondrion is metabolically flexible. It can easily burn glucose from carbohydrates, fatty acids from fats, or ketones from fasting to produce clean energy.
But in the modern world, because we're eating a high-carb, constantly fed diet, most people's mitochondria only ever burn glucose.
And that is a massive problem. When you force a mitochondrion to exclusively process massive loads of glucose day after day, it runs too hot. It produces a tremendous amount of metabolic exhaust in the form of reactive oxygen species free radicals.
It's like redlining a car engine constantly. Eventually, the engine cracks.
The clinical term for that cracked engine is mitochondrial dysfunction. And a cell drowning in the toxic exhaust of a broken mitochondrion begins to undergo massive genetic damage. The source material provided a deeply concerning statistic. Every single day, due to environmental toxins and normal metabolic stress, the DNA inside a single cell undergoes up to 500,000 distinct modification or damage events.
500,000 microscopic damage events per cell per day.
Yes. In a healthy, fasting-adapted individual, the rate of autophagy outpaces the rate of damage. There is a specific subtype of autophagy called mitophagy, which exclusively hunts down and digests these cracked, exhaust-spewing mitochondria, melting them down and replacing them with brand new, highly efficient engines.
But if you are constantly eating, mitophagy is turned off.
Exactly. In a diseased, chronically fed cell, the rate of DNA damage vastly exceeds the rate of repair. The broken mitochondria multiply, the cell fills with toxic exhaust, the DNA mutates, and that leads directly to severe pathology.
The infographic in the source material linking mitochondrial dysfunction to systemic disease was staggering. It obviously highlighted type 2 diabetes and insulin resistance because broken mitochondria can't clear glucose from the blood. But it also listed cerebral palsy, Parkinson's, epilepsy, cardiomyopathy, severe fibromyalgia, chronic fatigue syndrome, Huntington's, autism, muscular dystrophy, and Lou Gehrig's disease.
It underscores that mitochondrial health is the literal foundation of human biology. If the engines fail, the system collapses, regardless of what specialized organ it happens to be in. By utilizing fasting windows and AMPK activating spices, we force the cell to mercilessly cull the weak, broken mitochondria.
It's survival of the fittest on a cellular level, and the new mitochondria that replace them are vastly more efficient at utilizing insulin, which balances your blood sugar and stops the cravings.
And we must emphasize that this isn't just a strategy for reversing chronic illness. It is a profound optimization tool for highly active individuals and athletes.
Right, because intense exercise is fundamentally about creating microtrauma. You are intentionally causing mechanical stress and cellular damage to force adaptation.
Exactly. Intense training floods the muscle tissue with lactic acid, inflammatory cytokines, and damaged muscle proteins. By intentionally utilizing fasting windows and high doses of polyphenols like ginger and curcumin, athletes can drastically accelerate the clearance of that metabolic waste.
They are turning on the cellular vacuum cleaner post-workout.
Yes. The mitophagy generates fresh, hyper-efficient mitochondria that provide cleaner, more sustainable ATP production, leading to vastly improved endurance. And the active suppression of IL-6 by the gingerol significantly reduces delayed onset muscle soreness, allowing for faster recovery times.
Okay, I feel like my own brain has just undergone a massive process of neurogenesis absorbing all of this. Let's pull all the threads together.
Yeah.
So what does this all mean for you, the person listening right now?
It means that you are not a passive victim of the aging process. You possess an immense latent power over your biological destiny.
We started by looking at the microarchitecture. You have a built-in, highly advanced recycling plant inside every cell. The ULK alarms, the Vega-4 balloons, the lysosome incinerators designed to hunt down and digest the toxic, aging zombie parts of your body.
Exactly.
You manually turn this system on by simply pausing your consumption, whether that's through a daily 12-hour circadian window, a deep 48-hour therapeutic fast, or by biologically tricking the sensors with a fasting mimicking diet. You then pour rocket fuel on that natural process by flooding your system with the ancient chemical wisdom of polyphenols, ginger, turmeric, rosemary, and green tea.
And the compounding result of combining these biological hacks is profound. You forge a sharper, faster brain that is actively protected against the plaques of dementia. You seal and rebuild your intestinal wall. You clear your skin, and you erect a robust systemic defense shield against the inflammatory cytokines that drive chronic disease.
It fundamentally changes how we view hunger. It's an absolute paradigm shift from how modern medicine typically approaches health.
It truly is. Which leaves us with an incredibly important, lingering question to consider.
Let's hear it.
If human biology evolved over millions of years in an environment of scarcity, if we literally require periods of famine, and we require the ingestion of bitter plant compounds just to boot up our basic cellular maintenance software, what other modern comforts are accidentally turning off our innate healing mechanisms?
That is a thought to sit with. Next time you feel a craving late at night and you find yourself reaching for the pantry door, take a breath and ask yourself, is your body actually needing energy? Or is it begging for time to clean house?